All right. So we are now going to start on the topic of animals. So there's about 3 million to 10 million animals that exist on our planet today. However, these are estimates. So in reality, there's only about 1.5 million species of animals that have been described and given scientific names. So depending with new ways of analyzing data, there's probably more animals going to be, or there's certainly going to be more animals that are added to that total. So most of you guys are familiar with animals, so things like mammals and birds, but then there's other types of animals that you might not be so familiar with. So animals that don't really look like animals, like this sea anemone, right? Or other things like sponges. So things that don't really have like an obvious head or face. So animals originated from a single-celled eukaryotes, and they occur in a lineage that's called Opisthokonta. So this lineage includes the choanoflagellates, which are the closest living relatives to animals, and also fungi. So here you can see a phylogenetic tree that's showing the Opisthokonta. So we have the animals, their closest living relatives to animals, the choanoflagellates, and then another sister group, which are the fungi. So if you recall from the previous lecture, when we went over fungi, we did state that fungi are more closely related to animals than they are to plants and algae, right? So animals are eukaryotes that have certain key traits. They're multicellular. Their cells lack cell walls that you would see in plants and fungi, and they have an extracellular matrix within those cells. You also see that animals are heterotrophs, so they obtain their food from other sources of organic material, so from other organisms. Um, and really, what you see is that they ingest their food, okay, rather than absorb it. So when we looked at fungi, fungi are also heterotrophs, but when they obtain their um, those carbon compounds from other organisms, they did it by absorbing it. So they would release enzymes to break down the the large molecules into simpler compounds, and then they would absorb it. Animals just ingest their food. There are some exceptions. So some types of parasites do actually just absorb the food, okay? But for the most part, they ingest it. And the third trait is that animals have motility. So at some point in their life cycle, the animal the has the ability to move um, under their own power. So currently, biologists recognize somewhere between 30 to 35 different phyla. So these are the major lineages. Yeah. And if we look at these tables, you can see some of the number of described species um, of those phyla, within those phyla. Okay. So things like cnidarians, they have um, 11,500 species that are recognized to date. Porifera, which are the sponges, there's about 8,500 of those. Okay. And then when we look at more diverse groups, so things like the mollusks, Well, I'm actually blocking it, but for mollusks, there's about 85,000 different species of mollusks. And then there's the really diverse groups like arthropods. There's over 1 million species of arthropods that are recognized. Okay. Chordates, they're pretty diverse, so this is where mammals are. Okay, the class Mammalia is within the phylum Chordata. There's about 65,000 of these. But like I said, these numbers are not final, so they change throughout the years. So to study animals, biologists use several types of data. They use fossils, comparative anatomy, so comparative morphology. They use comparative development and comparative genomics. For fossils, these provide evidence of what ancient animals looked like, when they existed, and where they lived. However, there are some biases with these types of data because for fossils, these are more likely to occur for animals that were more abundant, so animals that had greater numbers, and also animals with hard parts like like sh- uh, like shells. Okay. So the fossils are also biased for animals that lived in areas where sedimentation was occurring, or that animals that lived recently. With comparative morphology, this distinguishes between shared characteristics and synapomorphies. So the data can be used to um, define the body plan of each lineage, to infer which characteristics arose first, and also to infer which animal groups are more closely related. The comparative development provides information about patterns of gene expression and morphological change during development. And then the fourth one, comparative genomics, this provides information about the relative similarity of genes of diverse organisms. And also, comparative genomics provides insight into the phylogenetic relationships and evolutionary history. So here you can see the phylogenetic tree that's based on DNA sequence data. Okay. So here, for each of these branches, you can see those morphological traits that distinguish the different types of lineages. So, for example, the platyhelminthes, which are flatworms, here this little line here on the branch indicates the loss of a coelom, because these animals are acoelomate, so they don't have a coelom. For annelids, like segmented worms, you can see that they have segmentation here, okay? Because this is a trait that is not present in mollusks. Okay? Same thing here for arthropods. So arthropods are segmented, but things like nematodes, which are roundworms, are not segmented. So here the little bars, that's what they're showing. They're showing those morphological features that are found in certain lineages. Okay? So the animals do form a monophyletic group. So if you recall, a monophyletic group is when you have a common ancestor and all of the descendants. So animals have a single common ancestor that was multicellular. And the origin of animals is based on data from fossils, comparative morphology, comparative development, and comparative genomics. So the prevailing hypothesis based on these data is that sponges are the most ancient lineage of animals. However, this isn't really saying that sponges are the ancestors of all animals. They're just saying that it's the most ancient lineage, right? Because sponges are just a sister group to the different lineages of animals, okay? Not the ancestor of all animals. Because if you remember when we went over the phylogenetic trees or the anatomy of phylogenetic trees, uh, all of these branches can be rotated. So just because you see a branch on top on a phylogenetic tree, that doesn't mean that that's where it's always going to be. They can be rotated around the nodes. And also remember that the branches can also be collapsed into smaller groups. So for example, the purple branches and the orange branches can be collapsed into a single branch called bilateria. Okay, so if you picture that collapsing uh, the purple branches and the orange into one single branch, okay, and then that single branch can be rotated with Cnidaria. So you can put Cnidaria at the bottom and that branch for bilateria in its place. Okay, same thing with Porifera. You can rotate Porifera down here and either all of these branches up here. Or if you collapse them, you can rotate bilateria up here. So sponges are just a sister group to the different animal lineages, not the ancestor of animals. So sponges um, do not have complex tissue. What they have is a group of similar cells that are organized um, into different functions. Okay, so here you can see an example of a sponge. So it doesn't really look like an animal, but it's an animal. Okay. Sponges, so sponges have no tissue layers, right? But there's animals that have two tissue layers. So these are known as diploblasts because these are animals whose embryos have two types of germ layers. So they have the ectoderm, which is what makes the outer skin or develops into the outer skin, and you have the endoderm, the inside skin. And then there's animals that are triploblasts. So these animals have three germ layers: the ectoderm, the endoderm, and the mesoderm. Okay. So here we can see a cross section of a cnidarian. So you can see there's only the two germ layers, the ectoderm on the outside and the endoderm on the inside. So the endoderm is the layer that would line some kind of central cavity here. Okay. So these are things. So diploblasts are like animals, are animals like cnidarians. Okay. So these are the groups of animals that includes jellyfish, sea anemones, hydras. So the germ layers develop into distinct adult tissues and organs. In triploblasts, the ectoderm gives rise to the skin, the central nervous system. The endoderm gives rise to um, the lining of the digestive tract, and the mesoderm gives rise to the circulatory system, muscle, and also things like bones and organs, or most organs. And so in general, the ectoderm provides the covering of an animal. The endoderm generates the digestive tract, and the mesoderm gives rise to the tissues in between. So again, things like muscle and organs. And then here's the two groups that have been recognized as diploblasts: the Ctenophora, which are the comb jellies, and the cnidarians. that I previously mentioned. Okay, so jellyfish, corals, sea pens, and anemones and hydras. So corals, if you look closely at corals, they kind of, if you zoom into a coral, you would see that inside of that big structure, that hard structure that looks very like a rock, inside of those, if you look at the little pores, you would see that corals are very similar to things like sea anemones. They're just tiny. However, there are some exceptions because there's recent data that suggests that some of the ctenophores do have mesodermal cells in their mesoglea. The mesoglea is just a middle layer between um, the endoderm, and the ectoderm. Okay, so it's a middle layer that's found in diploblasts. Okay, that's right in the middle between the endoderm and the ectoderm. And things like jellyfish, the mesoglea is mainly made of like water and mucus and different cells. It's what gives a jellyfish the squishiness. So there is some data that suggests that some of these ctenophores, some of these comb jellies do have mesodermal cells in their mesoglea. Okay, so we'll pause here and then we'll continue on the next video.